We're very happy today to welcome Raghu Mirmira, MD, PhD, and Sarah Tersey, PhD from University of Chicago. Today's talk is 12 lipoxygenase inhibition delays autoimmune diabetes in human gene replacement mice. And just a little bit about them. Dr. Mirmira is an international leader in type 1 diabetes research, specifically the biology of islet cells. His research is focused on the pathogenesis of diabetes mellitus and the biology of the islet. His laboratory is interested in three key areas: the role of mRNA translation in the cellular response of islet beta cells to inflammation, the role of lipoxygenases in dysfunction of beta cells in type 1 and type 2 diabetes, and the identification of biomarkers in beta cell stress and death in diabetes. Dr. Sarah Tersey is an islet biologist whose research focuses on the role of the beta cell in the development of type 1 and type 2 diabetes. The primary goal of her research focuses on understanding the molecular pathways that play a role in the development of diabetes, specifically pathways that include the enzymes 12-lipoxygenase and deoxyribonucleohypotensase synthase. By using inhibitors against these pathways or creating genetic deletions of either of these genes, Dr. Tersey has shown that diabetes can be prevented in mice. A second major focus of her research is the development of biomarkers that will best predict the likelihood of any one individual's risk to develop type 1 diabetes. Thank you so very much for joining us, and we look forward to this talk.
And the formation of neoantigens, which then get presented to the adaptive immune system. And then you start to see this picture of encephalitis. And then you can imagine that as the adaptive immune system enters, there is then a second phase involving interferons, such as interferon gamma, which then sort of augments this loop. So we think of this sort of process as a pathogenic loop. And that means that you can intervene at any, arguably any point, depending on the stage of disease, either intervene in beta cells, intervene in macrophages, intervene in the dendritic cells that present antigen to the adaptive immune cells, and you can intervene there as well. So this is part of the reason why you can intervene at different positions and still modify disease, and that's a direction that I think we're all thinking about, is intervening at more than one of these steps. But our lab has been primarily an islet lab, beta cell lab, and so we've been studying a variety of different pathways within the beta cell that get triggered by these cytokines, such as interferons. We know that they can lead through signaling to inflammation. They can lead to senescence. These can then subsequently cause oxidative and ER stress, which can then cause protein misfolding, formation of neoantigens, production of MHC class I, release of extracellular vesicles that communicate with the immune system, which then feeds back. You know, our interest has been to intervene in a variety of these different intracellular processes. And there are actually a variety of drugs that have either been introduced into clinical trials or have been tested in preclinical models that intervene in these different pathways. And some of these you're probably all familiar with. Our interest is obviously in the area of 12-lipoxygenase, and there's some others. DFMO, we published a study on that in humans about a year ago. So this is really the general approach of our laboratory, and that is that we study model systems, whether they are cell lines, primary cells, or preclinical models in vivo. And the angle here is identifying potential inhibitors or modulators of these stress responses, and then translating that to human systems, whether they be cell lines, primary cells, or in pilot clinical studies in humans. So what I want to tell you today is about a story related to something that we started in model systems and then developed a translational system that hopefully we can then actually move into a pilot study in humans. And that involves this enzyme, 12-lipoxygenase. I call it 12-LOX. It responds to inflammation, so a variety of pro-inflammatory cytokines can upregulate this enzyme. And what does this enzyme do? it takes arachidonic acid, which is derived from membrane phospholipids, and converts them into a variety of different metabolites that are inflammatory lipid mediators, such as 12-hydroxy-icosahexatetranoic acid, or 12-HEAT. And then these pro-inflammatory lipids can themselves transmit that sort of stress signal, activating ER stress and a variety of other pathways within the cell. And we think that some of these actions may be mediated through G protein coupled receptors. And then here are some papers in which we've published some of these concepts already. 12-lipoxygenase is important in human disease. And this is a study published by one of our collaborators, Jerry Nadler, who demonstrated that from NPOD tissue sections in pre-type 1 diabetic donors, you can start to see an upregulation of the 12-lipoxygenase enzyme in these presumably stressed beta cells. So, is that a cause or is that an effect? And that's really where our interests have lied. And so just as a very quick background study, we published this in Cell Reports a little over a year ago. And this was primarily led by one of my former postdocs, Annie Piñeros. What we did was we generated conditional floxed mice of the gene that encodes 12-LOX in mice. We crossed them onto the NOD background. And then we took a driver strain where we can knock it out inducibly within beta cells. And we crossed that onto the NOD background. And then when we deliver tamoxifen at the age that we choose, we can delete the 12-lipoxygenase gene in beta cells only, and then look to see how that modifies disease progression. And so what did we learn? So we delivered tamoxifen at an early pre-diabetic age in these animals, about 6 weeks of age. And then you could see a variety of different controls. These animals develop diabetes normally at the frequency you would expect for females and males. But the only genotype that was protected from diabetes were the animals that received tamoxifen and had the double LOX-P sites and therefore deleted the gene. And you could see here 90% of females were protected, 100% of males were protected. So clearly, knocking a gene out early in beta cells results in a reduction in disease progression. And if you look at the islet, it's really quite striking, is you could see in a control animal that there's a lot of invasive insulitis involving B cells, T cells, et cetera. Whereas in the knockout of the sort of beta cells in the of 12-lipoxygenase, that there was really a striking protection. So it's not like the beta cells were resistant to autoimmunity as much as it is the beta cells didn't transmit those signals to invite the influx of these immune cells. So that's what I would call disease modification. So this is kind of exciting. And then the question is, okay, we're not going to be knocking out genes in humans. Can we develop a drug against this enzyme? And so this was a study that had been years in the sort of development. We had a special NIH grant at the time to develop and identify inhibitors of 12-lipoxygenase. So we screened libraries. David Maloney was our collaborator at NCATS at NIH. Those compounds were sent to Ted Holman at UC Santa Cruz, where he did the biochemistry in vitro. Then those drugs were then sent to us, and we did human islet validation studies between myself and Jerry Nadler's group at the time at New York Medical College. And then the most promising candidates were then moved into preclinical validation in animal models. So there was a challenge, though, and that was that when we identified drugs, we found that there were 3 types of drugs. We could identify drugs that only inhibited the human enzyme, drugs that only inhibited the mouse enzyme, and then drugs that inhibited both the human and the mouse. So you can imagine this drug isn't all that useful in humans. We did publish that it does modify disease onset in mice. Well, great, but that's kind of a dead end. This drug category might be the most exciting, arguably, because you could test it in preclinical models and presumably move it into humans. It turned out that because of this lack of human mouse specificity, this category of drug turned out to be actually quite toxic. And so this was a big problem because it overlapped with a lot of kinases and phosphatases. So that left this drug, ML355, which was specific against humans. But the problem, as you can imagine, is it doesn't work in mice because it doesn't hit the mouse enzyme. The mouse enzyme is just sufficiently different than a human. So what that caused us to do over the years was generate a what we call humanized mouse, or a human gene replacement mouse, where we took the mouse enzyme and then we inserted the human enzyme in place of the mouse enzyme, left the upstream regulatory regions intact. So now wherever the mouse enzyme is produced, that's now where the human enzyme is being produced. And so this is really a model that can be used as a platform to test human drugs. So with that, I'm going to now turn it over to Sarah, who will tell us about the results of what happens when you give this drug to humanized mice.
All right, so moving forward, we're using these mice. So we back crossed the humanized mouse onto NOD all the way. And our control mice we have in the paper nicely show diabetes like we would expect in an NOD mouse. So the human enzyme nicely correlated with what we have in the mouse enzyme. And therefore, we made a great model to move forward. And so what we did is we took the NOD human ALX12 mouse at 6 weeks of age, and we treated them with vehicle, or what we now call VLX1005, which is that human ML355 that Raghu talked about. And then we treated them for 4 weeks, so from 6 to 10, and then we let them develop diabetes over the next about 3-4 months. And once they hit a blood glucose of 250 milligrams per deciliter twice, we consider them diabetic, or once they hit 25 weeks of age. Why am I going backwards? And what you can see here is that in both the females and the males, the vehicles confer diabetes at a very similar rate to what we normally see in NODs. So here we're about 75% in the females and then 50% in the males. And with VLX treatment, for just those 4 weeks, we see a nice delay that is longer than that 4 weeks. And it doesn't completely prevent it, but it does delay it out in both the females and the males. And then in order to understand what was going on and look at some of that mechanism and compare it to what we have seen when we knock out ALOX15 in the mouse, right? We did, oh, let me back up. Sorry, we also looked at if we can delay it, can we now treat it? And so these guys were allowed to get, we used the female mice, they were allowed to get diabetes. So they had a blood glucose over 250 milligrams per deciliter, and then they were treated with either vehicle or VLX. And you can see here in the blue that we had more mice that had a lower blood glucose for longer than we had with vehicle treatment. And then that's shown here with the average, and you can see we didn't really convert them to not having diabetes, but we did delay their blood glucose and keep them healthier for longer. And so now we're going to move back into the mechanism I was talking about. So we treated 6-week-old females with 4 weeks of vehicle and VLX, so like we did with that. But instead of letting them go and get diabetes, we went ahead and pulled tissues from them and started to look and see what was going on with the VLX treatment. And what we saw is here looking at in pink the T cells and B cells in this aqua color is that they have less encephalitis, very similar to our ALX15 beta cell knockouts. And that resulted in a decreased amount of encephalitis in all of the beta cells and all of the islets of the mice with VLX treatment. We see this at this 10 weeks of age. And if we use nanostring technology, that will allow us to capture a lot more information about what cells those are. And so we used this technology and we looked at the insulin area. So any area that's covered with insulin in green here. And then we compared that to the insulitic area, which you can find by looking at all the dense nuclei around. And we separated those out. And what you can see is looking at each of these different markers. So there's macrophage markers, there's B cell markers, there's T cell markers, is almost everything is down with VLX treatment. And so here's VLX treatment, and you can see that they're all lighter blue for the most part. All the way down, we have significance here next to it. So really VLX treatment is decreasing the amount of inflammation that we see at the islet at this 10 weeks of age time point. And then we looked specifically at T cells and macrophages using other stains. So we did a CD3 staining for T cells, just in general T cells, and you can see that there's less staining, both less bright, less dense around the islet, as well as F480 and looking at macrophages. In the islets that have less insulitis, they also had less macrophages within those islets, suggesting that those islets aren't going to probably ever have insulitis coming at them when there's so few macrophages here. And you can see that quantified here, they really had a lot less, both of the adaptive and the innate immune system. And so that's looking at what's happening to the immune cells around the islets. We also wanted to know what was happening to the islets themselves. And so we did RNA sequencing from islets that we isolated from the same 10-week-old time point, treated with and without VLX. And you can see that we got nice, clear separation. These islets are nicely different. And what we saw was a drastic upregulation of genes and not really a downregulation of genes, which was interesting to us. And you see some genes that are related to inflammation, as well as beta cell dysfunction and beta cell death and growth. And so here's the pathways that we found that were different after VLX treatment. These are growth pathways, and there's a couple that I wanted to point out, and that's a negative regulation of NF-kappa B activity, so inflammation, double-stranded break repair. So we're looking at beta cell death, and I'll show some data on that. Regulation of cell cycle transition, cell communication, and then immune response. And for the most part, if we go in and look at those genes, they're all down. And so we looked specifically at cell death by both tunnel and H2A.X, which is double-stranded break repair. And you can see that with VLX treatment, we really don't see any of these markers coming in that would signify that the beta cells are dying. And those are quantified down here. And additionally, when we look at proliferation in the beta cells using pCNA, or even in our nanostring that we saw before, which was Ki67, we saw a decrease of this proliferation. And we think that signifies a healthier beta cell. So in the NOD mouse, when the beta cells are dying a lot more, they're also trying to proliferate more to compensate. And we see both this decrease of dying and this decrease of trying to compensate for that proliferation, or compensate with less proliferation because they don't need to. So we, in general, think these beta cells are healthier. And to look at that, we looked at oxidative stress, which we have shown before that is 12-lipoxygenase is part. When we remove 12-LOX, we see reduced oxidative stress in high-fat diet models and NOD mice before. And so we looked at it here. And what you can see is a marker of oxidative stress. We have 4-H&E, and that's massively decreased with VLX treatment. And then GPX1, which is an antioxidant to resolve oxidative stress, is increased. So we have increased antioxidant with decreased oxidative stress that we're seeing in these islets, suggesting that they really are, in fact, much healthier than a normal NOD islet would be at 10 weeks of age. And then, so here we looked at total immune cells, we looked at the islets. And so then one of the other things that we have shown is that the 12 LOX is highly expressed in macrophages. And so we wanted to see what we were doing in those cells as well. So in order to do that, what we did is isolated bone marrow derived macrophages from our mice, and then we treated them. From the bone marrow, we proliferated them into macrophages, and then we polarized them into M1 macrophages, so pro-inflammatory macrophages using interferon gamma and LPS in the presence of vehicle or in the presence of 12 LOX inhibitor. And then we ran RNA sequencing. And you can see that after, with pre-polarization, there really isn't much difference in the M1, just non-inflamed macrophages. There's no difference. But once we polarize them, we see this difference between the VLX treatment and the vehicle treatment. And overall, there were a lot of common genes when we look from the M0 to M1 transition. But there were genes that were different between vehicle and VLX. And when we look at vehicle treatment, we saw a bunch of things related to inflammation and T cell activation and cytokines. And when we look at the VLX treatment, what we saw, oh, my things are off a little bit, but was negative regulation of type 1 interferon-mediated stress, cell response to virus. And these were all down. And so what we saw was this decrease of inflammation. And so they were M1 macrophages, but they were less inflamed M1 macrophages compared to vehicle-treated macrophages. And so in conclusion, I brought back the slide that Dr. Mirmira showed. And so we're here, right? And we think that really 12 LOX is playing this big role in both beta cell dysfunction and the inflammation that leads to beta cell dysfunction, as well as macrophage dysfunction. And when we inhibit it, we can decrease beta cell dysfunction, we decrease immune cell infiltration, and we decrease macrophage inflammation, leading overall to a healthier NOD mouse. And hopefully, because this is now a humanized mouse and a human drug, hopefully into a clinical trial, right? And we can show that we can do the same thing and lead to a less inflamed human islet as well, right? And so with that, just really quickly, Titley Nargis was our first author on our paper and did a wonderful job with this paper. And then the other people in bold are the ones, lab members that were also part of this, as well as our collaborators who Raghu mentioned. Jerry Nadler has been a collaborator with us for 12-lipoxygenase all along. Dave Maloney was with NIH, is now with Verilox, and is the one that provides us with all the inhibitor. And then Matt Boxer is also with Verilox. Thank you.
Thank you so much. That was fantastic. Just opening up for questions. Anyone? You can either unmute yourself or put something in the chat. Okay, I have a question. Do you guys see this 12 LOX working in this way in sort of other contexts or other disease contexts, specifically autoimmune contexts?
So mostly an auto-inflammatory context. Verilox Therapeutics was created just after we identified this drug. So in full disclosure, I'm on the scientific advisory board for the company. And it is not my company. It is Dave Maloney's company. But the initial indication right now that they have been pursuing, although type 1 diabetes is a major indication, and some of this work has been funded by Breakthrough Type 1 Diabetes, but their current indication is in heparin-induced thrombocytopenia, which, and the reason why that particular inflammatory disorder is because it turns out that 12 LOX, in addition to being in beta cells, macrophages, is also very highly upregulated in platelets. And so really quite, especially with heparin, these platelets get very inflamed, and then you start to get clumping of platelets and you get the subsequent thrombocytopenia. So it looks like in the clinical studies, this is really quite useful in that context. In other diseases, I mean, partly we need to look and see to what extent 12 LOX might be upregulated in the relevant cell types. We haven't. That might be something that the company is far more interested in than us.
And we have a question. You can unmute yourself.
Thanks. Hello to you both. Great to see
Hi.
you. I'm so excited about these studies. So thank you for sharing all of this with us, and congratulations. I'm just wondering, you did this short-term treatment, kind of pre-diabetes onset, and saw this protective effect. Do you know how long that lasts? Do you envision, when thinking about when in the course of the disease progression you would treat people? So do you think that this short-term treatment early on is basically reprogramming or retraining the immune system such that this would provide long-term protection? Or do you think that you would need to treat for a longer period of time?
So I think treating for a longer period of time is probably bad. Not that if we knock out 12 LOX in a normal regular animal, whether it was genetically or with VLX, we don't see bad phenotypes. So the mice don't stop growing or stop having, get sick or anything because they don't have an immune system. But I don't think the beta cell likes to have its dysfunction removed. Like, it needs its ER stress and its oxidative stress to keep producing insulin. So we're really proponents of short-term treatments. And we saw, with the diabetes treatment that we get a little bit of an extension. I really think it's going to have to be in a pre-diabetic person. So I had a lot of a positive type person like we have out there. It's possible it could go for a new onset or something. But we have not in our own hands done long-term because we don't like to inject mice over 4 weeks. For multiple reasons, but the mice don't like it either.
And I think, just to get to your question there, Maureen, I view this as disease modification, and that was a term I used a lot. And presumably, if we're modifying disease, you don't need to continually give a drug because you've modified the disease. So, if I had, visions of how would it be ideally provided, I would think in a pre-diabetic individual or high-risk individual who might need it only for a period of time that can get them out of, stage 1 or stage 2 disease.
Yeah, great, thanks.
So kind of like in a relapsing, remitting context.
Yeah, exactly.
Ernesto, did you want to ask a question?
Yes. Hi, Raghu. Hi, Sarah. The arachidonic acid pathway is very complex, has so many different products. Do you think there are other targets we can target?
I will say yes. We can target the upstream, phospholipases, for example. PO2G6 is a very good one. Ernesto, you and I have talked about that as a potential target. I think there are good and bad eicosanoids that emanate from these different lipoxygenases. In this study, which we didn't present, we actually did lipidomics to look at downstream, pathways that seem to be altered. It looks very much like mostly it's the 12 LOX products and mostly the 12 and 15 heats that seem to be most reduced. So, I mean, so I think, yes, there are probably other points in this pathway that may be prime targets. For example, what are the receptors for the heat?
I was going to say the receptors.
I think that those could be, even more, you can pinpoint, maybe.
How about the COX enzyme? Have you guys
COX?
ever? Yeah, COX.
We've not studied the cyclooxygenases.
Can I make a comment about that?
Yeah.
Please.
Yes.
So I think, there's, in addition to these heats that you guys are talking about, this is prostaglandins that come from arachidonic acid as well. And we've been working on some of the PGE2 receptors. And I think if you block the COX enzymes, prostaglandins do positive as well as negative things, and you're going to lose the positive effects as well as the negative effects. So I think moving more downstream and hitting specific enzymes or receptors is more the way to go than
I
blocking
see. Yeah.
high upstream.
I agree. I'm with you on that, Maureen.
All right. Thank you.
And I guess a quick question here. Can you talk a little bit about the impact of the gut microbiota on plasma oxylipin's profile, or specifically the 12 LOX profile, and how the change in the gut microbiome on the road to type 1 diabetes may intersect with 12 LOX, yes or no?
No idea.
So I don't know. And we've been giving this drug parenterally and not orally. Oh, were we giving this drug orally?
Orally, but we
Oh,
gave
okay.
it IP with the STZ and then orally, and we saw the same phenotype, but the NODs were oral.
Monica, I don't know. We're interested in the microbiome, and we're looking at it in a very different context. We've been doing some studies, and I think there's really something there. But and this is an interesting thought, right? Because we think that by hitting the disease early, we're reducing inflammation early. And could that be that we're reducing, microbiome-derived inflammation in some respect? And then are we modifying the microbiome? So, we just haven't looked.
Yeah, there's just a paper in Clinical Nutrition, April 2021. It was Torres Fuentes et al. And they're just going on kind of highlighting the significant correlations between different bacteria taxa and oxylipins, gut microbiota playing a key role in regulating the lipid metabolites and impacting inflammatory processes. It just was kind of curious because it seems like there's a potential, interaction.
Yeah, absolutely.
All right, then. I know you guys have to rush off to a HERN meeting, so thank you so much for everyone for coming. And if you could both just stay on for one more second as everyone exits the call. Thanks again, everyone, for joining.
Guys, thank you.
Bye. Thank you.
Appreciate it.
Bye.